Welcome to this video on protein synthesis. We will be going over amino acid structure, transcription, translation and proteins. Let's start by looking at amino acids. Amino acids are the building blocks of proteins. This image shows the basic structure of an amino acid. There is a carboxyl group on one side and an amine group on the other side. They also have side chains which are unique to every amino acid. So how are proteins made from amino acids? Protein synthesis has two major steps, transcription and translation. In transcription, DNA, a double-stranded double helix, is converted into mRNA, a single-stranded messenger RNA. Transcription occurs in the cell's nucleus. The enzyme RNA polymerase II helps catalyze the addition of complementary phosphorylated RNA nucleotides to the template DNA. Transcription occurs in three processes: initiation, elongation, and termination. In initiation, RNA polymerase binds to a region of the gene called the promoter. This causes the DNA to unwind and unzip. During elongation, the RNA polymerase slides along the template DNA strand, adding complementary base pairs. When RNA polymerase reaches the terminator region in the gene, transcription is stopped. The mRNA strand is complete and dissociates from RNA polymerase and the template DNA. Before mRNA can leave the nucleus, it needs to be modified. mRNA contains regions called exons and non-coding sections called introns. The non-coding introns need to be removed and modifications such as a 5' and 3' poly A tail are added. This is called intron splicing and is performed by a complex called a spliceosome. Then the exons are spliced together to form a mature mRNA molecule consisting of a single protein coding sequence. mRNA leaves the nucleus through a nuclear pore and attaches to a ribosome in the cytosol or on the endoplasmic reticulum, ready for the second process of protein synthesis, translation. Translation is the process through which mRNA is converted into a protein. This process determines the protein's primary structure. So how does translation occur? Translation, like transcription, is made up of three stages. Let's start by looking at initiation. The small ribosomal subunit moves from the mRNA methylated cap to the translation initiation site. At the start codon AUG, the tRNA with the corresponding anticodon will bind with an amino acid attached, in this case methionine. Once the tRNA is bound, the large subunit will then bind to create the E, P and A sites, with the first tRNA in the P site. During the next stage, elongation, a second tRNA with the corresponding anticodon for the next mRNA codon will enter the A site. A peptide bond will form between the adjacent amino acids. This is the beginning of the formation of a polypeptide chain. The first tRNA will release its amino acids, and as the ribosome moves up one triplet, it will exit through the E site and a new tRNA will enter the A site. This process continues cycling until termination. Elongation continues until a stop codon is reached, either UAA, UAG, or UGA. This will trigger the final stage, termination. There is no anticodon for the stop codon, leaving the A site unoccupied. A release factor will then enter the A site. The presence of a release factor will cause cleavage of the polypeptide from the P site. The final product is a primary protein ready to undergo further modifications. Protein structure. The primary structure of a protein is a simple order of amino acids joined together by peptide bonds. They form a linear polypeptide chain. A peptide bond is a covalent bond formed between two amino acids. At the molecular level, a peptide bond is formed through a condensation or a dehydration reaction. Two amino acids are able to bond together when two hydrogen atoms and an oxygen atom are removed from the molecules. One amino acid presents a carboxyl group to the reaction and loses a hydroxyl group in the reaction. The amine group of the other amino acid loses a hydrogen. The nitrogen then substitutes in place of the hydroxyl group, forming a peptide bond. The reaction produces one molecule of water. Secondary protein structure is the local folding of the primary amino acid chain. There are three main structures: the alpha helix, the beta sheet, and random coil, which is anything between alpha helix and beta sheet. The distinct structures are formed by hydrogen bonding within and between the polypeptide chains. The hydrogen bonds are formed between the amine group hydrogen and the carboxyl group oxygen found in the peptide bonds of the polypeptide chains. Tertiary protein structure is the 3D folding of secondary structures into protein domains due to side chain interactions forming non-covalent bonds. They can be comprised of any type of secondary structure. Quaternary protein structure is a functional and multimeric protein complex made of multiple folded protein subunits held together by non-covalent bonding. Today you have learnt about protein synthesis and the different protein structures. We hope you enjoyed this video. Thank you and goodbye.